Hi, my name is Alejandra Abramson, and my project is characterizing mesoporous carbon-based dopamine as an effective carrier for cancer treatment in HeLa cells. And this project was conducted in collaboration with the FIU Materials and Mechanical Engineering Department. So, cancer is one of the most deadly diseases that currently exists because there is no permanent cure. So, there are several treatments being conducted, which are chemotherapy and radiation, which tend to kill the healthy cells along with the disease cells, causing there to be an extremely high mortality rate because the patient gets worse before it gets better. About 9.5 million people are affected by countless variations of multiple strains of cancer, each and each getting more harder to detect. And it makes it one of the most widespread fields of research right now in the scientific community. So the strain of cancer cells that I use are HeLa cells, which are Henrietta Lacks cells cultivated in 1951 because of their advantages. They are cervical cancer cells that are widespread and have several commonalities with several variations of other cancers or within the same type of cancer. And they divide extremely fast, making them testable within days of exposure. So, nanoparticles are a brand new field in materials research because of their applicability to be filled by any sort of chemical or treatment. So, they can enter the body nasally or orally and are activated using light, meaning that extreme light can cause a heat release of chemicals that were within the nanoparticle. So, these are the six most wide known benefits of targeted treatment, which are cell targeting because it can only affect a certain tissue and won't affect the healthy cells, DNA modification because it can change the DNA of the tumor cells so that when they replicate, they'll replicate healthy cells instead. Controlled release because they only release the dopamine or whatever chemical is inside whenever designated by the heat concentration given, and the variety of administration techniques, which is either nasally or orally. And engulfment basically states that they can be absorbed by a certain cell, but not another, meaning that the healthy cells don't have a chance of receiving the chemicals that the targeted cells do. And fill determination basically states that different chemicals can be inserted into the hollow center of the nanoparticle, making it a widespread treatment for different types of diseases, not just cancer. And nanoparticle research has one base idea, which is that they are carriers because they can be hollowed out and filled with treatments or chemicals. And the two main uses of these nanoparticles are medical imaging, which they can be an effective carrier because they can carry radioactive isotopes to test for certain functions, and gene and drug therapy, which can target certain cells for diseases or DNA codes that can prevent certain diseases from being passed on or getting worse. So, mesoporous carbon-based dopamine focuses on three certain characteristics in order to be the most efficient carrier. They have pores that allow them to be controlled by heat release in order to release the dopamine into the cell. They can be targeted because of the different types of chemicals that they can carry on their hollow inside. And the carbon that makes up their structure provides a proper protection for when the nanoparticles enter the body and enter the cell so that they don't break down. So I focused on one characteristic, which was heat, and did two different types of property testing in order to test its efficiency. So I tested for stability, which is the stability of the cell and of the nanoparticle within the cell to prevent breaking down or death, and it limits the possibility of malfunctioning release. And the retinins test how much the cell can hold of the nanoparticle and still continue to divide and accept more. So, my hypothesis was that if mesoporous carbon-based dopamine is introduced into cancer cells in increasing concentrations, then the higher end of the variable spectrum will have a higher mortality rate and a less targeted effect. And then the purpose of this experiment is to utilize the photothermic effect of mesoporous carbon-based dopamine for biological application of cancer treatment cells. And so my procedural timeline states that after the research, experimentation, assays, and data collection is done, and then the analysis is conducted in order to confirm significance and the validity of the data. So these are my main materials, which included the HeLa cells, the mesoporous carbon-based dopamine, the media that promoted cell growth, the 808 NIR laser that was provided by the lab, and the CCK8 assay, which tested for cell viability after exposure. So the experimentation basically stated that 80% of the HeLa cells had to be confluent on the plate. So there had to be an 80% concentration, and there were 4 trials per plate. And then there were 10 variable concentrations across, and each plate was done at least 3 times for each concentration. And then the goal was to introduce different concentrations of the mesoporous carbon-based dopamine into the well plate in ascending doses of double the one before. And since each trial was done multiple times, it could assert the validity of the data, and the setup is shown as below. The 3 assays that I did were the cell viability, the photothermal, and the photostability assay. The cell viability assay tested for the survival rate of the cells once exposed to the mesoporous carbon-based dopamine in increasing concentrations. And then the photothermal assay, as shown below in the picture, uses a laser to test to which extent the mesoporous carbon-based dopamine is affected and can absorb the light without breaking down or having a malfunction release, which is releasing the dopamine without need. The photostability assay tested for the range at which the laser kills the cells or decays the mesoporous carbon-based dopamine because of excessive heat and can prevent treatment release. So this is the IC50 data. It relies on the analysis of the gradient that is shown. So the control at the very beginning shows how there was a no level of absorbance because there was no mesoporous carbon-based dopamine, but as the level of absorbance of the particles increased, the gradient did get darker. So as shown in the graph, the 200 nanograms per ml did have a high absorbance, but tended to kill all the cells, meaning that it was a much darker than all the rest of them. So this is the cumulative IC50 results, which show that there were little to no outliers in the data. And the data presented that the 100 nanograms per ml tended to be the most accepting while also having a high concentration of mesoporous carbon-based dopamine without decaying or killing the cells. The photothermal assay also supports that concept and that conclusion because the 100 nanograms per milliliter was the most effective and was able to absorb lots of heat without breaking down. So the conclusion and analysis states that yes, 100 nanograms per milliliter was the most ideal concentration of nanoparticle. And it also validated that mesoporous carbon-based dopamine is a valid drug-carrying treatment for cancer. Since dopamine is the most widespread chemical used to treat cervical cancer, this was the most applicable in this area. And that it also showed that target-specific treatment is a valid concept and valid idea and can be used in the future for other types of cancer research. And these 3 different factors that were considered were the manipulation of the structure, the inner lining, and the type of cancer being affected. And then the data analysis did show that all data collected was statistically significant with very few to no outliers depending on the assay. And that the 3 different types of assay presented 3 different types of data to support all types of conclusions regarding the data. So there's a vast application for this project across the entire medical research community because it will work to improve the future of cancer treatment, because the medicines will become less harsh on the patient and will only allow them to get better because it will not kill the healthy cells, only the targeted tumor ones. And since healthy cells will be saved, it will have a lower mortality rate and more people have a higher chance of surviving. And then some future research for this experiment is that there will be different types of coding structures with different chemical fields to try to test certain different types of cancer or diseases. Or the nanoparticles can be used as a plasmid vector, meaning they will carry DNA into cells in order to change the DNA structure so that when they replicate, they are no longer diseased. And that different types of lasers on a different type of spectrum can have a vast range of effectiveness. So purple light may be more effective than the light given by the 808 NIR laser. Thank you. If you have any questions, please direct them to the document attached. Thank you.